So in this presentation we're going to solve problems using Punnett squares for monohybrid crosses. So the dot point specifically talks about monohybrid crosses using Punnett squares or other appropriate techniques. The other appropriate techniques could be the cross method that we used in the Mendel presentation. So before we get started, let's revise a little bit of our terminology. So we're just going to go through some of the words that we've used so far. So genotype. Now genotype refers to the alleles present. So it's the alleles present. And it's often talked about in terms of homozygous and heterozygous. So the other word there is the allele. So what is an allele? Well, an allele refers to different forms of the same gene. Okay, so for example, we looked at plant height in Mendel's pea plants. So the different forms of the same gene for plant height. So height is the gene that is responsible for plant height, and the different alleles are tall and short. Okay, so let's move on. Next word is homozygous. So homozygous genotype is a genotype where both alleles are the same. So it's a purebred individual for that characteristic or that trait. Okay, so we can refer that to again, homozygous tall. So what that means, if we use our letter notation, is that both of the alleles are the tall variety. So it would be TT, remembering that there are always two factors, or he talked about two factors or two alleles for every gene that are present in an organism. The other word is, so we've got homozygous tall, and we could also have homozygous short in terms of pea plants. Now that would be little t, because the short was the recessive. So if homozygous means that they're purebred, homo meaning same, heterozygous refers to it being a hybrid. So it's a hybrid, and both alleles are different. So again, if we're looking at plant height, we would say heterozygous, and because heterozygous has got one of both, the dominant being tall, we would say heterozygous tall. That is its genotype, and we would write the genotype in our notation as big T with a little t. So the tall we've found out is the dominant gene, the little t is the recessive gene, and both of them have to be present in a heterozygous individual for that trait. Note also that we always write the capital first. Capital must be written first. So the last word we're going to look at is this term phenotype. Okay, so phenotype just refers to the physical appearance, what we can see. We can't always work out what the genes that lie below. That can be a bit of a lottery. We don't always know what's there, but we can talk about the physical appearance, what we actually see. So in Mendel's dwarf plants, you could see that they were either tall or short for height. So the phenotype is the physical appearance, just what we can see. So we're going to use the Punnett square to solve the following problem. So it says in Labrador dogs, black fur, capital B, is dominant to white fur, little b. Predict the expected genotypic and phenotypic ratios resulting from crosses between a homozygous black and white dogs. Now, to solve this problem, we don't just jump straight into a Punnett square. We need to go step by step, and I expect that every single time we do crosses that you take these steps. This will make sure that you don't make any problems in determining what the outcome is. There is no mix-ups in what the parent genotype is. So let's start off with step 1. So step 1, we need to designate letters. Provide letters that are going to tell us information about their genotype. Designate letters. Now, there are a couple of tricks to designating letters. We always obviously use capital for dominant and lowercase of the same letter for recessive. Now, the reason why we use the same letter is if we use different letters, we could be looking at 2 genes. We're only looking at monohybrid crosses, so we're looking at the same gene, we use the same letters. Okay, so last thing to say here, it's always good to use letters that are able to tell the difference between their uppercase and lowercase. Like in this example, they've used B for the dominant, and they've used little b for the recessive. We can see there's a clear difference between them. If we start using letters like C, and we've got our lowercase c, we can see, yeah, there's a difference between them. But if you're filling in your Punnett squares later on, you might get those confused. You might draw one a little bit bigger and one a little bit smaller, and it gets very confusing to tell what the outcomes are. That's where mistakes can be made. So B's, A's, D's, and there are a number of letters which are good letters, and there are others like S's, C's, which can be very confusing. O's would be another one, because their upper and lowercase look very similar. So that's step 1. Okay, so for the example here, we need to designate letters, and this has been given for us. So we know that dominant is B, dominant is capital B, and that is black fur. And recessive is lowercase b, and that is white fur. So, step 2, write down the phenotypes for the parents. Write down phenotypes for parents. Now, so let's look at this example. We call this our P1, our parent generation, and our P1 genotypes. Now, this one's pretty clear. It tells us in the answer what we're looking for. So it's a homozygous black dog, black being dominant, so it is a capital B. We know that, we've got our letters there. So our parents are a homozygous black dog and we're going to cross that with a white dog. Now, white fur is little b. That tells us little b is recessive. For the dog to be white, both of its alleles should be little b. Step 3. What we need to do in step 3 is list all the alleles. So list all alleles. Each parent can pass on to gametes. Okay, so list each allele, each parent can pass on to gametes. Now, this is also important that we show segregation is occurring in this step. Okay, so we have our genotype. So this is before segregation occurs to form our gametes. after segregation occurs, so we have segregation in this step, and we now have our gametes as they separate, and we just put them a little bit further apart, B, B, little b, little b. So the next step is to, so step 4, draw a Punnett square. This is to help us find the combinations that are going to exist. So we'll just get a Punnett square in there. So now we've got our Punnett square here, we're going to put in our gametes. So we can place these guys here in these squares on the side, and these ones here up above. Now it's a simple task of making crosses. Okay, so we were looking at this square, what goes in this square, we look at the square to the side and above. So it would be a capital B for the dominant black and a little b for the recessive white. Move over to this one, again we see the one to the side is a capital B and the one above is a little b. And we repeat this down the bottom, knowing that the same pattern is occurring. Capital B, little b, capital B, little b. So step 5, so we write out genotypic, phenotypic ratio. We note that that it is asking us for a ratio, and sometimes it might ask us for a percentage. In this case, it's asking for a ratio, so this is what we're going to provide for them. So first of all, so this is our F1 generation. This is the outcomes of the Punnett square cross, so the genotype. So in this case, they are 100% or they are all heterozygous black. Phenotype, again, they are all